So, as I mentioned, today I'm going to focus on the value of always validating and optimizing your assays for whatever your particular research model in question are. First, I just want to give a brief overview of the Preclinical Research Center, which is my research team. We are directed by Dr. Mel Graham, and we are in the Department of Surgery at the U of M. And we focus on developing methods to study immunometabolism and other complex diseases. Specifically, we focus on non-human primate research. We also use other research models, but that's really our specialty. And there we go. We have a really wide breadth of research, and this is just to give you kind of a little snapshot of the types of research that we take part in. We have a large transplantation program because we are in the Department of Surgery. We work closely with the chief of transplantation at our local hospital. We do solid organ transplant, both allo and xenotransplant for kidney and liver. We also do isolated cell transplant, so islet cell transplantation in diabetic models. In addition to the islet transplantation, we do just a lot of type 1 diabetes research that focuses on a variety of different therapeutics to help improve outcomes for type 1 diabetic patients. We also look at metabolic disease and metabolic syndrome that can be comorbid with obesity. We've done some vaccine development, specifically during lockdown. We did work on some COVID vaccine studies with our primate models. We also do some device testing, regenerative medicine, and gene therapy studies. So again, sort of our major goal is just to conduct preclinical trials with the utmost rigor and reproducibility to be able to move towards FDA approval and application to the clinic. So model development is incredibly important for what we do. We do exploratory and discovery studies that we perform non-GLP, but we still follow established standard operating procedures that outline the proper operation of equipment, maintenance, sanitation, reporting structures, and all related activities. So we call these GLP-like. But we also do IND or IDE enabling studies that can be conducted fully GLP, so we have that capacity. We have a really mixed portfolio of funding from federal institutions, state institutions, foundations, and industry awards. And we do our own research as well as partner within our own university, with outside academic institutions, and with industry partners. So a really nice, diverse portfolio, so we're never bored. So when you think about choosing a model for your research, you want to look at the different types of model validity and what will best answer your research question. So typically, if you're sort of at the beginning of researching a therapy, you might want to use in vitro or in silico models to look at phase validity. So that's usually screening models that will mimic the physiology that you're looking at, such as hyperglycemia or glucose intolerance. Usually once you make it past the screening, you want to look at construct validity, and that's where you get into your small animal studies. And some large animals like pigs and sheep also can work for construct validity. And those are models that will have a similar underlying biology or etiology of the disease that you're studying, but they may not reflect like the immune complexity that humans do. So then finally, when you're moving to predictive validity, as in you want to see how well a therapy will function in the clinic, you want a more rigorous and stringent model, which is where NHPs usually come in. And these are models that will have a similar response to humans, to clinically effective and appropriate interventions and therapeutic agents. And they also have the immune complexity to really model what you might see in the clinic. So why do we choose primates? So we all know that in vitro and rodent studies generally support pathway determination and mechanistic studies, which is very valuable, and it's always where we have to start. But NHPs are used to examine the practicality, safety, and generalized efficacy of any therapeutic that has a really high relevance to the clinic. And this is due to their close genetic relationship with us. They have anatomical, behavioral, developmental, psychological, and reproductive similarities to us. And especially as I work a lot in transplantation, the similar immune system and the complexity of their immune system is very important for really testing in a stringent model different therapies that might work because they have similar MHC expression and high level of conservation in amino acid sequences and complexity. And there's also a lot of cross-reactivity with human-directed biologics and susceptibility to infections, the same as humans. So this really allows us to create a clinical trial-like preclinical safety and efficacy study that can parallel the therapies and improve translatability to the clinic. So where cytokines come in for us is a couple ways. So cytokines, as we know, are critical regulators of inflammation and immunity. And being able to assess them, especially in phase in a study, is very important for us because we often have long-term studies. And I also use the multiplexing approach as a method of refinement in research. It allows me to assess a large number of analytes using a very small blood volume, and which reduces the burden on the animal, because we're always trying to reduce their burden and make it as best of an experience as we can for them. And we can also, this is something I'm going to get into in a few more slides, we can access our animals cooperatively, so we don't have to sedate them because they've undergone cooperative training. So this is really a nice method of refinement for us in our research that still has, that allows us to get at different indicators of health, disease progression, and treatment efficacy depending on the study. And it helps us monitor responses to a test article, whether it's a device, gene therapy, a biologic, and obviously it's a target for immune manipulation in the case of transplant. So it's very highly valuable for us. So how can we implement cytokine analysis with scientific rigor and reproducibility in mind? So a lot of labs don't necessarily do this, but something that I always like to sort of preach about is always doing your own in-house validation of any commercial kit, because no matter how much work that a company does to validate their kit, it's still not done in your hands with your exact model and your exact variables. It's always just going to put a little bit of difference on it. So here's something that we've published that's just an example of a validation that we did. And this was a number of years ago, but we were validating a multiplex cytokine kit. And we did this by taking serum samples from 20 different cynomolgus macaques, and we validated the cytokine assay across 4 different plates. And we looked at a number of different outcome measures. So we looked at recovery, so that's the observed concentration relative to the expected concentration, and we measured samples neat and then spiked with different kit standards. Then we have acceptance criteria for all of these outcomes, and within this multiplex kit, only 13 of the 23 passed for our model. We then looked at linearity, which is the observed concentration relative to the expected concentration in a sample with a known dilution. And we spiked it with a known concentration of a standard and measured it neat and at dilution. And again, only about a little more than half, 14 of 23, passed. We then looked at precision, both intra and inter-assay precision, and all of these analytes passed in this kit except for TNF-alpha on inter-assay. We also looked at sensitivity just to see if the reported lower limit of detection was accurate in our hands, and in this, all of them passed. So we take all that information and put it together into kind of a matrix and see which ones pass on all counts. So we had 11 that met all of our acceptance criteria, 7 were outright rejected, and 5 passed some but not all criteria, so we consider them marginal, which means we can still use it, but you have to take it with a little bit more of a grain of salt. So where it gets interesting is if you think about that matrix we created, we also looked at within subject variability. So we took samples from several animals across multiple different time points, and then measured the cytokine values, which these were all naive animals and nothing was happening to them, so it should be pretty much the same. But here you'll see the cytokines that had the highest variability within those time points are the ones that also corresponded to rejected validation measures. So this is something that if you hadn't done this work, you might be accidentally attributing what you see as a change in cytokine expression to your test article or your disease model, but in fact it's just an analyte that's a little bit more variable for your application. So that was, as I said, a number of years ago, and that's this first competitor kit where we had only about 40% of analytes regularly falling within detection range and about 40% of our inter-assay CVs within 5%. So then we partnered with R&D, Biotechny, and they said, hey, do you want to try our kit? And we said, yes, we would love to. We have an NHP kit. And initially, we had about the same issue with analytes not falling in the detection range, but our CVs were really tight. So we worked with them a little bit, so we have a partnership, and we optimized the assay performance. So we tried a few things and ended up increasing incubation time to improve binding, and we expanded the number of points on the standard curve so that we could get more at those lower concentrations. And with that optimization, we now have a kit where we have 83% of the analytes falling in detection range, and our CVs are almost 100% in range. So this is pretty much our standard kit that we use now for cytokines. Next, I want to talk about, in addition to getting the correct kit and understanding how it performs in your hands, looking at some of the intrinsic variables that can influence your scientific outcomes. So these are demographic variables, such as species. So in our case, we use both cynomolgus and rhesus macaque, which have some inherent species differences. Their background, so NHPs are outbred. It's not like your inbred mice strains. And sex. Preclinical and clinical studies both tend to be male skewed. And then there's, of course, things such as age, disease status, origin, things like that. So we have this published as well, that we did find there are sex differences in cynomolgus macaques in just their baseline cytokine expression. So if you don't have an equally balanced cohort in your study, you might again be getting some influence there if you're only looking at males or only females, or if for whatever reason your population is just skewed one way or the other. And then something that we focus on a lot at PCRC is our model-imposed variables, all of which can lead to increased stress in the animal, which will then, of course, affect all of your outcome measures. So these can include your experimental design, which might include restraint of the animal, the timing of any interventions you have, and the frequency of interventions or blood draws. The severity, is it a very severe disease model? Is it invasive, surgical model? Is there a large cumulative burden just because there's a lot of sampling time points? Their environment, is it adequately enriched? And are there species-specific needs? Like, does the animal need to forage, or are they social? And that gets to sociality as well. Are they single-housed, pair-housed, or gang-housed for social species? All of these will affect the stress of the animal, which will have a high impact on any of the analytes that you're looking at. So some of the physiological effects of restraint and stress, they can have direct activation of the HPA axis, which will change your immune parameters and inflammation downstream. And there's also chemical restraint, which is commonly used, and this can introduce a direct drug-related physiological effect, especially on your cytokine expression. So restraint-based sampling of macaques is very commonly used. It's FDA required. And this will include sedation or physical restraint of the animal. And this obviously creates a lot of stress for the animal, or chemical confounding if you're using ketamine or other sedatives. And this is obviously less representative of the clinical situation. We aren't always sedating our patients to take a blood draw or holding them against a wall. Like, that's just not what happens. So what we specialize at PCRC is cooperative training of our animals. This is less commonly used because it is a time investment, but we are able to train our animals to participate in the research that they do so that we don't have to sedate them. They know what's going on more or less, and they're just calm and less stress. So this leads to less potential confounding of our outcome measures, and it's obviously much more representative of the clinical situation. So here I'm presenting some work from one of our grad students, Scott Oppler. And here he's looking at model-imposed effects of research outcomes using a single event chemical restraint. And here he found these are animals that had a single sedation event with ketamine, and he compared those with those that were cooperatively sampled. And you can see here, so the red is the cooperatively sampled and the yellow is our sedated animals. And this is just a single sedation event. and there was nothing else. These were all healthy, normal animals. You see this significant blunting effect. That again, if we didn't know this effect, you could attribute it to your test article or your disease. But in fact, it's just sedation that's having this blunting effect. And so think about that, and then think about, these are just some examples of some very common dosing sampling timelines that we see in the NHP world. So the top one is a 42-day safety efficacy study that has, multiple sampling time points. Across those 42 days, there's a 24-hour PK study where there's multiple samplings within 1 day, which can be really intensive for the animal. And then what we really specialize in is these disease management studies, such as diabetes models. So that's diabetes induction, and then we usually handle the animals twice a day to monitor blood glucose, give them insulin, give them food. It's just highly intensive on the animal. So not having to sedate them makes all the difference in the world, both for them, for your research ethics, and for your data. So here is a study that we had. So we did have an FDA study that was requiring us to sedate animals for sampling, and we used that as an opportunity to say, what is this repeat sedation? What effect does this have on some of our parameters that we commonly look at? So we paired some of these animals that were getting chemically sedated to our cooperative animals. So we had 6 in each group, and then we wanted to look at incidences of AEs as well as cytokine expression. And you can see here that the chemical restraint-based sampling resulted in increased adverse events, specifically vomiting and nausea and inappetence, which are always things that the FDA is looking for when you're testing any new intervention. And when we used that chemical restraint, we had a significantly higher rate of vomiting and nausea and inappetence in these animals. But our cooperatively sampled ones, we had hardly any incidence of that. So here's where it gets very interesting. So then we looked at these animals that had repeated sedation events. And again, so the yellow here is our cooperative groups, and this is looking at cytokines over time in this 42-day study. And you can see that all their cytokine levels were rock solid, like they just did not move. And then when you look at the ones with repeated sedation, you see that first time, so like it repeats what we saw with a single sedation event. You see an initial blunting of your cytokine expression, but then with repeated sedation events, it actually increases. So this is an effect that a lot of studies, if you're using this sedation, would inaccurately attribute to some other part of your experiment rather than stress that's imposed by your model and the effect of chemical restraint potentially. So it's really important to understand what these variables are and how they're impacting your research. Now I'm just going to get into a couple other neat ways that I use cytokines in my work. Our animals are also instrumented with ports so that we can draw blood from them easily. So they have a vascular access port, and sometimes we'll do a portal port. And this allows us to interrogate things between compartments in the same animal. So here, this is again our healthy animals where I just looked at the difference in cytokine expression between portal and peripheral blood. So that was just a neat little pilot study I did. And this is something that is under review and hopefully will be published soon. But we have completed a study with an implantable device for type 1 diabetes. And this is a device that operates as a bioartificial pancreas. And it has a cell house. So this is the whole device that you see pictured here gets implanted subcutaneously. And the cell house is like a little chamber where we can then load it with islet cells. They could be porcine islet cells or stem cell derived ones. It really doesn't matter. They can all go into the cell house. And then there's a pump in the middle and an accumulation disc, which is furthest to the left in that diagram. The accumulation disc will collect interstitial fluid, and the pump will pump the interstitial fluid through the cell house to sort of feed the cells, and then it will secrete insulin out of that. And so what we wanted to know was what kind of cytokines are the transplanted cells expressing, and then is that having an impact on the systemic inflammation. And hoping you can see it well enough, but I had to split the axes because it was so different. So the blue bar is what I measured from inside the cell house that our islets were secreting, but you can see that the yellow line, the systemic circulation was pretty quiet and nothing was happening. So even though there was this really high concentration of cytokine expression within the device, it wasn't having any kind of adverse effect in the animal as a whole, which was great. Oh, my next slide. There we go. So in conclusion, accurate interpretation of preclinical data is dependent on understanding your model-imposed variables, such as restraint you're using, sedation, cooperative handling, anything like that. The intrinsic variables of your model, species, sex, these are things we can't control, but we just have to account for. And your analytical accuracy, do you fully understand how the assay performs in your model and in your hands, and how that might affect any of your measurements and outcomes? Multiplexing cytokines is a key refinement in the use of NHPs. We're always trying to reduce the amount of blood we have to take from them and do the most with the least amount of samples. So multiplexing is great for that. And the pathway level analysis gives us a view into health status and disease progression, as well as how any interventions are performing. The qualification and optimization of any commercial kit is critical. It increases our translational accuracy, and it improves translation into the clinic, and sharing our refinements via open access publications is just something I always am a cheerleader for, because it just leads to improved science across the board. So I'm just gonna leave you with a summary. Again, you can screenshot this if you want. This is our group, this is our director, Dr. Mel Graham. So we have a full clinical arm that can run studies, full behavioral management of our animals and support. We do intensive surgical models, disease modeling, cutting edge clinical care, and anatomic. We have pathologists on staff. And then the in vitro service is what I head up in PCRC. And so I handle and direct all of our cage site analysis, which includes hematology, chemistry, et cetera, sample collection processing, controlled SOPs for everything. And we have a full comprehensive QA system. So you can reach out to us if you have any interest or just want to chat. We're always open to new collaborators. And finally, I just want to leave an acknowledgement to our funding sources, our incredible team at PCRC, most of which are pictured there, and some of our PI partners. Any questions?